Welcome to this lecture on the neurobiology of behavior. Today, we are going to explore a fascinating question. How does our physical brain create our complex, everyday actions? We will look specifically at how we process language and how sexual orientation develops. By the end of this session, you will understand the biological machinery that makes us who we are. Now that we have introduced our topic, let us start by thinking of the human brain as a highly advanced biological computer. Just as your laptop requires both physical hardware circuits and specific software code to play a video, your brain needs specific neural pathways to produce complex behaviors. When you learn a new word or feel romantic attraction, it feels completely natural and automatic to your conscious mind. However, beneath the surface, billions of neurons are firing in highly specific, genetically and hormonally guided patterns. We're going to explore exactly how this biological hardware creates the complex software of your everyday behavior. Building on that computer analogy, let us define what the neurobiology of behavior actually is. It is the scientific study of how the central nervous system translates genetic instructions, hormonal signals, and environmental inputs into complex actions and cognitive processes. It specifically examines the neural circuits and anatomical structures governing functions like language and sexual orientation. For instance, language relies on specialized regions in the left hemisphere to convert abstract thoughts into spoken words. Similarly, reproductive behaviors are deeply influenced by early fetal exposure to hormones, which permanently organize the developing brain. Understanding these biological foundations leads us to an important question. Why does this field matter so much? Primarily, it is absolutely crucial because it removes the stigma from neurological and psychological differences by explaining them as natural biological variations. For example, if a stroke patient loses the ability to speak, knowing the exact neural circuits involved allows doctors to design targeted speech therapy. In the realm of sexual orientation, recognizing the biological and hormonal underpinnings helps society understand that attraction is a deeply ingrained, neurologically programmed trait, not a simple conscious choice. This knowledge bridges abstract psychology and concrete neuroscience. Now that we see the real-world impact of this science, let us look at how we measure these biological influences, starting with genetics. To truly understand how strongly genetics influence a specific behavior, like a language disorder, neuroscientists rely on a mathematical concept called heritability. This concept allows researchers to separate the effects of a person's DNA from the effects of their environment and upbringing. By quantifying these differences across a large population, we can see exactly how much of a behavior is hardwired into our biology. This brings us to the mathematical formula that expresses this relationship. This concept of separating DNA from environment gives us the heritability formula. Conceptually, it compares the genetic variation to the total observable variation in a population. Let us break down the symbols. H squared represents the heritability estimate, which is a proportion between 0 and 1. V sub g stands for the genetic variance, meaning the differences caused strictly by genetics. V sub p represents the total phenotypic variance, which includes both genetic and environmental influences. If the genetic variance increases while the total variance stays the same, heritability approaches 1, meaning the behavior is almost entirely genetically determined. Before we move on to hormones, let us see this formula in action with a concrete example. A team is studying children with specific language impairment. We are given the total observable variation, or phenotypic variance, as 150 units. The variation due strictly to genetics is 90 units. We are asked to calculate the heritability. We choose the heritability formula to find the exact proportion of the behavior that is genetically driven. We substitute 90 for the genetic variance in the numerator and 150 for the total variance in the denominator. Dividing these gives a final answer of 0.60. Physically, this means exactly 60% of the variation in this language disorder is due to genetics, leaving 40% to environmental factors like education. Having seen how genetics shape behavior, we must now explore another massive biological influence: hormones. Beyond genetics, the physical structure of the brain is heavily shaped by chemical signals called hormones during early fetal development. These hormones do not just float around. They must physically bind to specific receptors in the brain to trigger the growth of behavioral circuits, such as those governing sexual orientation. To measure how effectively these hormones activate the brain and organize future behavior, neuroscientists use a specific equation derived from biochemistry. Let us look at how we calculate this hormonal activation. Following our discussion on fetal brain development, we arrive at the receptor binding equation. Conceptually, this formula predicts the strength of behavioral organization based on the amount of hormone present. Let us define the variables. B represents the amount of hormone bound to receptors, which drives the behavioral response. B max is the maximum possible number of binding sites in the tissue. The letter H in brackets represents the concentration of the hormone circulating in the bloodstream. Finally, K sub D is the dissociation constant, measuring how tightly the hormone binds. A lower value means a stronger fit. If the hormone concentration increases significantly, the binding approaches the maximum limit, leading to a highly pronounced behavioral response. Now that we have our receptor binding formula, let us apply it to a scenario involving fetal development. We are given a maximum receptor capacity of 500 picomoles per milligram. The dissociation constant is 20 nanomolar, and the fetal testosterone concentration is 80 nanomolar. We are asked to calculate the bound receptors to see if they are more than half saturated. We use our formula, substituting 500 for Bmax and 80 for the hormone concentration in the numerator. In the denominator, we add the dissociation constant of 20 to the hormone concentration of 80. Multiplying the numerator gives 40,000, and adding the denominator gives 100. Dividing these yields a final answer of 400. Since 400 is well over half of the 500 capacity, this intense hormonal signaling will strongly masculinize the neural circuits. Having calculated how circuits are formed, let us visually examine one of these fully developed circuits in the human brain. Here, you are looking at a lateral view of the brain's left hemisphere, which is highly specialized for language. Notice the two highlighted regions. In the frontal lobe, we have Broca's area, labeled for speech production. In the temporal lobe, we see Wernicke's area, labeled for language comprehension. The large curved arrow connecting them represents the arcuate fasciculus, a bundle of nerve fibers that allows these two areas to communicate. This diagram teaches us that complex behaviors like language are not handled by a single spot, but by a network of specialized physical structures working together. Just as we saw the physical layout for language, let us now look at the microscopic mechanism of hormonal organization we calculated earlier. In this schematic, you are seeing exactly how a chemical signal alters the brain. On the left, the molecule labeled hormone physically locks into the receptor on the cell surface. This specific event, labeled binding, is exactly what our previous equation was measuring. Once bound, an arrow shows the process moving to signal transduction, where the message is passed inside the cell. Finally, this leads to the physiological response at the bottom, which in our context is the permanent wiring of behavioral circuits like sexual orientation. This visualizes the direct link between chemistry and long-term behavior. To wrap up our lecture, let us review the most important ideas we have covered. First, we learned that complex actions, from language to sexual attraction, are deeply rooted in physical brain structures and chemical signals, acting much like a biological computer. Second, we saw how mathematical models, like heritability, allow us to quantify exactly how much of a behavior is driven by genetics versus the environment. Third, we explored how fetal hormones permanently organize these neural circuits through receptor binding. You have now covered the fundamental biology of behavior. Always remember that recognizing these biological bases helps us appreciate the natural diversity of human experience and develop better medical care.